In the previous lecture, we finished with coenzymes and cofactors. We looked at the structures of some coenzymes. As for the cofactors, we saw them to be metal ions. We even looked at mechanism of action of the coenzymes and also the sources where we get them from. I mean, they are generally derivatives of some vitamin Bs. Today we'll continue to look at factors that affect enzyme activity. And then we'll take a look at mechanisms of action of enzymes. Take a look at some mechanisms. We'll refer to some specific examples. So hopefully, over a period of 1 hour, we should be done with what we have for today. And if you have anything to say, if you have any questions, you have any clarifications, the floor will be open for you. The chat is open, so if you have anything, you can write in the chat. I'm monitoring it, I can see it. Or alternatively, you can note your questions down, your comments you'd like to make. You can note them down, and at the end, when the floor is opened, you can put up your hand. I will enable your microphone, call your name, then you will unmute and speak, and I'll respond to you. Okay, so let's dive in. We are going to take a look at 4 major factors that influence enzyme activity. The first is enzyme concentration. The second is substrate concentration. The third, temperature. And then the last one will be pH level of the environment in which the enzyme is acting. So when we take enzyme concentration, if you look at the chart which is on the left-hand side of your screen, we have a relationship between enzyme concentration and the rate of reaction. And clearly, the rate of reaction increases with increasing concentration of enzyme. So there is that direct proportionality between the rate of a reaction and enzyme concentration. Of course, that is all things being equal. When you have other conditions like temperature, pH, substrate concentration, all of these things are kept constant, and enzyme concentration is being varied with increase, then that will also result into increasing rate of reaction. I mean, this is understandable because you have enzyme molecules that are proteins, and these enzyme molecules will be acting on substrates. So if you have some amount of substrate there and you have enzyme molecules that are proteins that should be acting on this substrate to be forming products, the more enzyme molecules you have, the faster the reaction proceeds, because then it will mean that more substrates will be converted per unit time to a product. So you will have such proportionality between the rate of the reaction and the enzyme concentration. The second is substrate concentration. So here too you have in the beginning you see that the rate of reaction increases with increasing substrate concentration. Then it gets to a point that increase in substrate concentration does not result in any change of the rate of the reaction. At that point, we refer to as a saturation point. Now, why that? Why that? So let's see. So if you look at the diagram that is on your screen right now, there's a reaction where enzymatic reaction, the substrates are the small spherical, pink figures, whilst you have like the bean-like structures. These are the enzymes that will be acting on the substrate. Now, in the beginning, you see that you have a small amount of substrates. You have more enzymes there, and you can see that some of the enzymes are free. They are not engaged by any substrate that they will be acting on. So you will have just a few of these substrates that the enzyme will be acting on. Now, in the second phase, you see that the amount of substrate has been increased. And when the amount of substrate increased, you see more of the enzymes are getting occupied by substrate molecules. Of course, this is going to result into increase in rate of the reaction because you are now having more substrate upon which enzymes are acting. You will have more products that will be formed. In the last stage where you are seeing high substrate concentration, you have more substrates than enzyme molecules. And what you see is that all the enzyme molecules are engaged. They are all acting on some molecule of substrate. And you still have extra substrates with no enzymes attached to them. At this point, you have reached a saturation point so that no amount of substrate that you add into such highly concentrated reaction mixture will result into any change in the rate of reaction, because the amount of enzymes that you have in the reaction concentration, the reaction mixture, will only act on the amount of substrate that it can act on at a point in time. Until it finishes, it is not going to act on another molecule. So whatever you do, adding more substrate, you are not going to have faster products. You are not going to have products coming out faster. This is what we saw at the saturation point earlier. So you see the curve plateaus. You see the curve, first of all, in the beginning is increasing. The rate of reaction is increasing with increase in substrate concentration. Then it starts to plateau all the way and flattens out and gets constant. So that at this point, any amount of substrate that you are adding will not result into a change in the rate of the reaction. That is a saturation point. So you have such saturation curve that results in change in concentration of substrates. The third is temperature. Temperature. So how does temperature affect the rate of chemical reaction, biochemical reaction? Now we see, usually we have, if you take the human body, for instance, our physiological temperature is 36.6 degrees Celsius, let's say approximately 37 degrees Celsius. So it is at this point that you have all the enzymes in the body being optimally active. And generally, in many organisms, they have physiological temperatures. I mean, then physiological temperatures actually they hover around these 37 degrees Celsius, except for maybe some thermophilic bacteria that are living in deep sea that can live in conditions even up to 100 degrees Celsius. Of course, those ones, they can have their enzymes in those conditions acting optimally in such very high temperature, such very high conditions of temperature that supports, their life. But here, I mean, you see generally that optimal temperature, let's say that the optimal temperature is like 37 degrees Celsius. And you observe that when the temperature increases above that 37 degrees Celsius, the rate of the chemical reaction reduces, which means the enzyme activity reduces. Of course, we have said from the beginning that enzymes were mostly proteins. And so that should give you an understanding that if you increase temperature above a certain level, it's going to result into denaturing of the proteins. And once the proteins get denatured, they lose their functional structure. And once they lose their functional structure, they will no longer be able to carry out their function of biocatalysis of the biochemical reactions. So obviously the rate of the reaction will reduce all the way until the reaction no longer, takes place because all the enzymes are destroyed. They are denatured by high temperatures. I mean, health-wise, you know what it means when a body temperature rises above, let's say if you have a body temperature 38, 39 degrees already, clinically, people will be moving to put that temperature under control, because what it means is that if it continues to rise, there will be breakdown of proteins, and particularly when you start having breakdown of blood proteins. Let's say hemoglobin starts to break down. That, of course, will be resulting into some anemic conditions. And then all the other proteins that you can think of, apart from the enzymes, hormones, you can just imagine what will be happening. Things like nucleic acids begin to break down. That is in very, very severe conditions. Some of these things will result even into fatality. How about when you consider the reduction in temperature from that optimal temperature? You see, when the temperature is below the optimal temperature, there too you see that the rate of reaction is also reducing all the way until the rate of the reaction actually gets to zero. Now, what accounts for this? You see, enzymes, yes, as proteins acting at an optimal temperature. Reduction in temperature below the optimal temperature also reduces the activity of the enzymes. The enzymes become less and less inactive when the temperature is lowered below the optimal temperature. It is actually based on such knowledge that we have preservation of food, for instance, by refrigeration. Or even sometimes, those of you who are working in microbiology laboratories and things like that, you do store some of your, whether samples or reagents in the refrigerator. I mean, food. So if you take food, for instance, I mean, you are preserving food in the fridge and maybe freezer for what? In order for the food to be kept for a longer time. You are preventing it from spoilage. And that is because, I mean, a lot of times food will get spoiled when you have microorganisms that are acting on the food. Let's say the biomolecules that are in the food, like carbohydrates, lipids, proteins, breaking them down because those microorganisms are also feeding on those things. And then their byproducts become toxins to us because those are released into the food. The byproducts are released into the food and the food becomes toxic so that after when you take it, you get things like food poisoning. So you want to put your food inside a refrigerator so that you reduce the activity of the enzymes of these microorganisms that will be acting on the food. In that way, you can keep the food for a longer time. It doesn't mean that the enzymes or the microorganisms will not continue to be acting on the food. In this case, remember, the proteins are not denatured. When it's a higher temperature, the proteins get denatured, and so they no longer act. But when it is lower temperature, you only reduce the activity of the enzymes. But those microorganisms, if you bring that food back into a condition, say to the optimal condition or a temperature above that low temperature, which is in the fridge, like maybe 5 degrees Celsius or anything, the rate of reaction will increase. So even if you keep food in a fridge, it can still get spoiled. It's just a matter of time. It's just that the rates at which the microorganisms act on the food is slowed down so that the food can be kept for a longer time. And so even for instance, when you buy some packaged foods, for instance, you have them being labeled like expiry date is written on it. But then there is also an information that when you open it, you should consume it in maybe 24 hours or 48 hours, 72 hours. They give a certain period of time that you should consume it. And that's because when a food is packaged, it's sealed, a lot of times hermetically sealed, meaning away from oxygen. So at that condition, that anaerobic condition, microorganisms that are existing already in the food will not be acting at a high rate. But the moment you open it, you'll expose it to air, you expose it to oxygen. And so the rate of chemical reaction by these microorganisms will increase, which will mean you have to consume it before the microorganisms grow up to a point that it becomes toxic to you when you consume such foods. So yes, temperature, has these two ways. Either when you reduce the temperature, it reduces the rate of reaction. the enzymatic reaction, or when you increase it, that will result into denaturing of the enzymes and will also lower the enzyme activity. But in fact, if you increase it, what it means is that you have destroyed the enzymes, and so the reaction will not even continue. It's just that as you are increasing it, more and more of the molecules of the enzymes will be getting denatured, which will be resulting into reduction until all the enzymes get denatured and the reaction no longer proceeds. So temperature is one of those things that can be employed for inhibition of enzyme activity. Now pH. pH also, the influence of pH looks also very much like that of temperature. In this instance, different enzymes have their particular pH levels at which their activity is optimal. So, for instance, if you take, say, alpha amylase in the mouth, so we have this salivary amylase in the buccal cavity that acts on carbohydrates. And in the mouth, pH levels are somewhere around neutral, slightly basic. But in the stomach, for instance, you have the acidic condition. So what it means is that the enzyme that is active in the mouth will become inactive in such acidic condition in the stomach. But there in the stomach too, you have enzymes, such as pepsin, which will be acting on protein, which means that enzyme is active at such pH level, such acidic condition, but it's not at a pH level in the mouth. Or maybe in children, in babies, that you have some enzymes like renin, which will be converting milk proteins into thick curds, things like that. So you have different enzymes with different pH levels at which they operate optimally. Even that if you have the pH levels at very extreme ends from the optimal pH, it can also result into denaturing of these proteins, just like how temperature does. In this case, it can be both ways. If the pH is lowered or the pH is increased at very extreme ends, this can also result into denaturing. And such a denaturing is a breakdown of the protein. So the enzyme will become inactive altogether. So the reaction is completely terminated. So that's about major factors that influence enzyme activity. Right. Now we can take a look at some mechanisms of action of enzymes. We take a look at two major mechanisms. The first is the lock and key model, which is also referred to as the Fisher mechanism. And the second is induced fit model, also referred to as the Koshland mechanism or Koshland model. Now, the first, which is the lock and key model, was proposed by this a German chemist. I think he came up with the proposal that enzymes and their substrates actually have a fit between them. There is fitness between them, such that the enzyme has a portion. We'll come to the, I think when we're looking at terminologies, we mentioned what an active site was. So that active site of an enzyme, has such a fixed shape, which fits to the substrate. So that is substrate specificity to an enzyme. So a substrate must, perfectly fit to an enzyme for an enzyme to act on it. So you see on the right-hand side, what is depicted here is that you have a substrate, which is red, and the enzyme is blue. And if you look at the active site is having a certain shape, which is sort of fixed. That's how it is. It's fixed, fitting exactly to the substrate for the substrate to fit in it to form an enzyme substrate complex. Now, in fact, Fisher actually got a Nobel Prize for his works with enzymes, particularly with this model. Got some Nobel Prize for that. So we see that is depicted more for you to see that you have a substrate. So the substrate is, depicted like a keyhole, and you have the enzyme, which is the key. So that's why it's called a lock and key model. Mostly you hear it's being referred to as a lock and key model. Many may not mention the name Fisher to it, but this is the person who proposed that. So sometimes too you can see it being referred to as a Fisher mechanism or the Fisher model of enzyme action. So we see the substrate and the key fit exactly tight together. So that model, is showing that you have an enzyme molecule, and this enzyme molecule is having some active sites into which you can have some substrate binding. So these are some substrate molecules, the black sphere, and then you have some square with a groove in it. And these have some specific sites in the enzyme that fits to them. So you have a site that looks round, which will fit to that sphere, and you have a site which also will fit to that square with a groove in it. Now we see those two substrates now bind to the enzyme to give you an enzyme substrate complex. And you can see right away that there is some interaction between the two substrates in the active site of the enzyme there. And that would then result into formation of a product. So we see the product at the end, which is separated from the enzyme. So the enzyme is free. You now have a free enzyme that can continue to act on more substrate molecules that are present, and you have your free product. So that was the full proposal from Fisher. But considering such a proposal, what it means is that if, for instance, we take, say, what you see, so we're starting from the top, A. That's a depiction of a reaction without an enzyme. So a non-enzymatic reaction, a reaction which should occur spontaneously. And that is depicted with, say, if a substrate is, let's say it's a metal stick, that's a substrate, and that metal stick is to be broken into two. Those will be the products. So if the product is to be broken metal sticks, two of them, and we consider that the transition state of that metal stick, that substrate, is its bent state. So you see the metal stick is bent. What it means is that it's first of all gotten to a stage where it can be easily broken, because once the metal is bent, there is that dent in it, and so it can easily be broken into the two sticks. So just an analogy to show that. Now, with the proposal from Fisher, what it means is that this metal stick, if we take the enzyme to be a magnet, because according to Fisher, the substrate fits perfectly to the enzyme. And so it means that it's like a magnet that fits perfectly to the metal stick. So the enzyme, which is depicted here in blue, having this magnetic field, which is perfectly fit to the substrate. And you see the enzyme substrate complex is formed when you have the substrate bound onto that enzyme as a magnet. Now, you take your mind back to when we were looking at the energy profile of both enzymatic and non-enzymatic reactions. We took a look at energy profile of chemical reactions such that we had substrate at a certain energy level. And I mentioned to you that if you take reactant molecules that are at an energy level, those reactant molecules, they exist as those compounds, for instance, because they have such stable states. And so in order for them to react, there was the need for activation energy to take them to an energy level which was unstable for them. Then they can begin to react to form a product which will be at a lower energy level where it will be stable. You remember that conversation on the energy profile. So if that's the case, then it means that if you have an enzyme which perfectly fits to a substrate, then the enzyme substrate complex would be so stable that there will not be any need for that enzyme substrate complex to proceed to form a product. That's why you see that the reaction ends the moment you have the substrate fitting so perfectly to the enzyme. It will mean that already has become a stable energy level state, that the reaction will no longer proceed. That enzyme substrate complex then becomes the product. And so the idea was modernized to show that actually what should have been is that you have a substrate that is not perfectly fit. It's not perfectly fit to the enzyme, but after it binds to the enzyme substrate complex, which is formed, now has a state already, because in that state you have the substrate being bent. The enzyme can now break it so that you now have the product that is getting separated from the enzyme. So the first idea of Fischer was built upon by Daniel Koshland. He's an American biochemist. So he built on the first proposal by Fischer to say that actually the enzyme is flexible, it's not fixed. So he brought some the idea of dynamism. The enzyme is dynamic, so that it isn't that the active site is fixed, fitting a substrate, but rather when the substrate binds to the enzyme, it results in the active site of the enzyme changing to fit the substrate, meaning the active site of the enzyme is adaptable. It's not fixed. It's dynamic. So it can change to fit the substrate to now give you that enzyme substrate complex. So that's where the name came from, induced fit. So what it means is that, according to Koshland, if you take a substrate, so that's a substrate violet, and you have the enzyme with a cream color with some active site being shown. When the substrate enters into the active site, it induces the enzyme so that the enzyme now would change its shape to fit the substrate. Now, that then results into the full mechanism by what Koshland was referring to as the induced fit model. So here we see this is an enzyme yellow that is having an active site being shown, to be round. I mean, just to depict what Koshland meant. And you have a substrate, and the portion of the substrate that should bind to the enzyme, you see, has a square shape. But in the presence of the substrate, the enzyme then assumes some conformational changes. So there are some changes that occur in the conformation or the configuration, or say the structure of the enzyme, so that now the active site of the enzyme is changed to fit the substrate, so that now you now have an enzyme substrate complex that then will result, so that's a transition state, the enzyme substrate complex, that will result into formation of products that then get dissociated from the enzyme. So that was Koshland building on the idea, the proposal of Fischer. And actually, this is the modern understanding of how enzymes act, to know that enzymes are dynamic, they are flexible, they are not. Even though they are specific to their substrate, you have such substrate specificity, but you also know that you have some enzymes that are group specific because we did classification of enzymes. So some enzymes will be acting on different substrates. So of course, this model fits better than the first model by Fisher, because if you take the first model of Fisher, what it meant was that every single enzyme was going to be substrate specific. It will not act on any other substrate except one. But this knowledge of the induced fit model will fit for enzymes that are also group specific, bond specific, and even we looked at other specificities like stereospecificity, for instance, or specificity pertaining to the coenzyme that is acting. All right. Now we can go back to what we refer to as the active site of an enzyme. And we mentioned that the enzyme, which is a big protein, is having a specific portion where the reaction that it is catalyzing will be occurring. So it's not that such a big molecule protein, any portion of it, anywhere at all, then there's reaction occurring on it. We mentioned that from the first lecture. So if you take this big structure as a protein that is an enzyme, the blue portion is the active site. So that is where the reaction should be occurring. It's like a pocket. It's like a pocket inside the protein, because the protein is folded. And inside that pocket there, the reaction will be catalyzed. And that's where the reaction will occur. That's where the substrate will bind. And so we see a substrate molecule, which is depicted here, black molecule has entered into that active site. Then we also have some, portions which are being shown red as the substrate binding site. What are those? So you remember we mentioned that, you see, a protein is made up of amino acids. And we looked at the primary structure of proteins, which was the sequence of amino acids in the protein chain. So you have some specific amino acids that when the protein folds, these specific amino acids form that active site. What it means is that it is these specific amino acids that act directly with the substrate. So a particular point of those amino acids are going to be interacting with the substrate. And that's what we are referring to as the substrate binding site. Those specific amino acids, some specific portions of them that are having some chemical interaction with the substrate. Those are the substrate binding site. Now, in the active site, generally, there are some common amino acids that you see. These are histidine, cysteine, aspartate, also known as aspartic acid, arginine, glutamate, also known as glutamic acid, and serine. So you mostly come across these amino acids in the active sites of enzymes. Now, if you pay close attention, you'll realize that these amino acids that we have mentioned, have some acidic or basic side chains. So here again, I would say that you should be going back to take a look at the structure of the amino acids and pay attention to the side chains. So for instance, if you take aspartic acid, take a look at the structure of aspartic acid. We did these things even in the HND level. So you have those things. And I think even in the first lecture, we did some quick revision. I think I gave you a slide with the structures of the amino acid. So you can quickly use that and refer to, say, if you take aspartic acid, for instance, what's the side chain? How about glutamic acid? What's the side chain? You see that you have some extra carboxylic acid groups on the side chain. That is apart from the carboxylic acid group, which is on the alpha carbon. You have an extra carboxylic acid group, which is on the side chain. So that would be an acidic side chain. Arginine, histidine. There you see some amine group. Histidine has, I think when we go forward, we may see an example with histidine that you see that histidine is having an imidazole ring structure. Histidine is not considered as an aromatic amino acid. It's not an aromatic amino acid. That ring is not aromatic. You see, it's some azo ring. You have some nitrogen in there. We'll look at it and you see it. So those will be acting like basic. Then you also see serine. Serine is having a hydroxyl group on it. And so that hydroxyl group can have some interaction with substrates, even with water, forming some hydrogen bonds with substrates. We'll see, some reaction mechanism where serine is involved in some enzymes that act on proteins. And then cysteine. Cysteine, too, you remember, cysteine is having a sulfur hydral group, one of the amino acids that has sulfur in it. It's a small amino acid having SH there. And so that sulfur hydral group can also, give out hydrogen atoms, or let's say a proton or hydrogen ion. So you see, a lot of times what you see is that the activity of these enzymes is based on some acid-base reaction, what we refer to as acid-base catalysis. You see that, so some transfer of proton, hydrogen ion given out, and then one amino acid receives hydrogen ion. And so once it receives a hydrogen ion, it is acting as a base, something like that. That's going to result into the activity of the enzyme. All right. Now, let's take an example of, say, an enzyme papain, or you say papain. So now we are looking at the active site of the enzyme papain. And actually, papain actually it's It's an enzyme that acts on proteins. It breaks peptide bonds, and is actually used in the meat industry as a meat tenderizer. So that is some practical application there for you. So if we take the green part, the green, where you are seeing the green part of this diagram as the protein pipeline. So it's a protein chain. It's long, it's large, it's big, but it folds. And as it folds, we see in the active center cysteine and histidine. Now, interestingly, there are some numbers there. Cysteine 25, histidine 159. I think I explained I explained this in one of the lectures to tell you that, look, it's just that you have a protein that is having amino acids that are, joined with peptide bonds, giving you a peptide chain. The protein is large with these amino acids in it, and if you are considering the primary structure of these proteins, you have cysteine as the 25th amino acid. When you are counting the amino acids from the first all the way to the last that are involved in that big protein. And histidine will be in the 159th position. These cysteine and these histidine molecules in that chain are the proteins, are the amino acids that act in the active center of the enzyme. And you see, there's a protein which is being depicted here, just showing the peptide bond. And a peptide bond is formed between a carboxylic acid group of one amino acid and the amine group of another amino acid. The rest of the chain is shown with R and R prime. So the bond which is to be broken is between the carbon and the nitrogen. That is the peptide bond that is to be broken. And you see the sulfur in cysteine is having some lone pair of electrons that can have some nucleophilic attack on the carbon. And you see a hydrogen that is on that amine group that is involved in that peptide bond, also having some hydrogen bonding with a nitrogen in the imidazole ring of histidine. So this is the imidazole ring I was referring to. So it's sort of putting a stretch on that peptide bond, and in the end, that peptide bond will be broken. So that is the action of papain on peptide bonds. Now, if you take, what we have, spoken about so far, and you want to look at, so how do enzymes, carry out their action on substrates? We are taking the mechanism of enzyme reaction such that if this is an enzyme, so this is an enzyme having an active center. So I explained to you the active center is like a pocket and it's supposed to act on, say, substrate. So we have substrate A, substrate B. So here you are having free enzyme. You have two free substrates. And these two substrates are to react between each other. They should be reacting. And the enzyme is to catalyze the reaction between these two substrates. Now, how will that occur? How will such a action occur? So from left to right, we are looking at the mechanism from left to right. So the first thing you see is that you have a free enzyme and the substrates. Free enzyme with the substrates. Then you see the substrates now bind in the active sites of the enzyme. So you now have the enzyme substrates complex. Enzyme substrate complex is formed. In that enzyme substrate complex, in the active center there, you now see the substrates react. So the substrates in the active center, they react. The enzyme, of course, we saw earlier that it catalyzes the reaction by reducing the activation energy of these substrates. So the reaction occurs there in the active center and products are formed. And then these products are released. The enzyme is free. So if you have an enzymatic reaction mixture where you have substrates with the enzyme in there, you have molecules of the products and you have molecules of the enzyme. So if we take, for instance, that usually you have more substrate molecules than enzyme molecules. So when you have the enzyme catalyzing the reaction of just one molecule, product released, other substrate molecules can also bind until the reaction. So that reaction will continue to be catalyzed with products forming until all the substrates, the reactants, are consumed in the reaction mixture, and you'll be left with just the products and free enzymes. All right. Now, that's well understood. We can now continue to look at some specific example. So we take chymotrypsin, which is an enzyme also involved in the breakdown of peptide bonds in proteins. And chymotrypsin is classified as a serine protease. You see, we saw that in the active center, one of the amino acids you find is serine. And in many enzymes, you have in the active center serine performing an activity, which is a proton transfer, which results in the activity of the enzyme, or it makes the enzyme active. So when an enzyme is inactive and is to be activated from serine, a hydrogen from the side chain, that hydroxyl group, OH group on serine, is given out, which is referred to as a proton transfer. You will see that in many enzymes, and those enzymes are classified as serine proteases. So we are looking at the serine protease mechanism, taking chymotrypsin as an example. Now, before I talk about this reaction mechanism, let me just mention to you that, please, this is not a thing that you want to go and be cramping into your head. No. just understand the step-by-step process by which this is occurring. So it's not a thing you cramp into your head, but you just try to understand what it is that is happening, arrow by arrow, reaction by reaction. That's all. Now, we see this is a serine residue. So there's a serine residue there in the active center. And we also see histidine. Histidine. So you are seeing histidine here again. When we're looking at the example with papain, we saw cysteine and histidine. Here we are seeing serine and histidine. And you notice that again, that nitrogen in the imidazole ring of histidine is involved in receiving proton from the serine molecule. So you see, right from the beginning, there is that interaction being shown between that hydrogen of the hydroxyl group in serine with that nitrogen in the imidazole ring of histidine. The substrate, again, is shown here. That's a protein. The peptide bond is between the carbon and the nitrogen. The rest of the chains, the rest of the peptide chain is shown with R. So this is the bond that is to be broken, the bond between the nitrogen and the carbon. That is the peptide bond to be broken. Now what we see is that this hydrogen from serine, from the hydroxyl group, is donated onto the nitrogen of the imidazole ring of histidine. So you now see the hydrogen is on the nitrogen. Then that hydrogen, which, has been donated onto that nitrogen, is now having an interaction with the nitrogen involved in the peptide bond. Again, that will be resulting in some stress on that peptide bond. And you see there is that, temporary covalent bonding between the oxygen of the serine and the carbon also of the peptide bond. So that's an intermediate that is formed there. You have the protein which is to be broken down, now having, such complex formed with the amino acid residue, serine and histidine in the active center of chymotrypsin. The next thing you see is that part of the protein, the N-terminus, where the nitrogen is in the peptide bond, is now broken off. It's released because the hydrogen is donated. The hydrogen that histidine accepted from serine is now donated onto that nitrogen. And that part of the protein molecule is released. And you see the carboxylic acid end, the C-terminus, is still bound to the serine residue. The next thing is that water comes into the reaction. That will be some hydrolytic reaction to occur. First, the water molecule gives, is having a hydrogen atom given onto that nitrogen again on histidine. And you have the hydroxyl group which will be, because water, there's a hydroxyl group, which is left. That hydroxyl group now goes to the carboxylic acid group, the C terminus, which is bound on the serine. So you see that hydroxyl group in that intermediate, that hydroxyl group is gone onto that carbon. In the end, what you see is that the hydrogen from the water which was accepted by histidine goes back onto the serine molecule. It's like the serine molecule has gotten back the hydrogen that it lost from water. But first it went to histidine before histidine, transferred it onto that serine molecule. And then the OH from water now goes onto the C terminus of that peptide bond which was broken, so that you now have a complete carboxylic acid group. And so that part also gets dissociated from serine. And you now have the original form of the enzyme back. This is the inactive form of the enzyme that, for it to get active in the beginning, the serine gave the hydrogen to the nitrogen on histidine. So it's called proton transfer. It's a mechanism by which enzymes get activated to carry out enzymatic reactions. You see it in many enzymes as a proton transfer, activation of enzymes by proton transfer. As I mentioned from the beginning, please don't try to go and cramp this in your head. It's just for us to follow step by step what happens, for you to appreciate the fact that, yes, you write chemical reactions and say reactants and then arrow product. But as a matter of fact, there are step-by-step processes that occur. It's fast, but these are mechanisms by which these reactions occur. All right. You are not doing a biochemistry major, so don't worry so much about this. Then mechanism of action of lysozyme as another example. We take a lysozyme. This time around, it is with carbohydrates. And lysozyme is what we refer to as glycoside hydrolase. So we classify it as a glycoside hydrolase that's involved in hydrolytic breakdown of carbohydrates. And which bond will these hydrolases break down? They are going to break down glycosidic bonds between either monosaccharides or monosaccharide derivatives of carbohydrates. And lysozyme is one of such hydrolases. So hydrolase was one of the classes of enzymes that we saw. And we're looking at 6 classes of enzymes, referring to oxidoreductases, transferases. The third one was hydrolases. Before we went forward, talk about lyases, isomerases, and ligases. Now, lysozyme, you find it, virtually in all our tissues. Lysozyme is naturally produced because it's needed for our innate immunity. You find it in like the tears coming from our eyes, in the tissues. You'll find lysozyme in mucus. You'll find some lysozyme there. And it has this antimicrobial activity And the mechanism by which it acts is to, break down a polysaccharide, which is referred to as peptidoglycan. And peptidoglycan is a polysaccharide of monosaccharide derivatives and acetylglucosamine. So what you see, NAG, what you see here, NAG is N-acetylglucosamine. And NAM is N-acetylmuramic acid. So again, from HND first year, looking at carbohydrates, monosaccharides, and monosaccharide derivatives, you must have, done these things. Those of you I taught first year HND, obviously, we did these things. So if you've forgotten, you can go back and take all those first year things and look at N-acetylglucosamine, N-acetylmuramic acid, which you see them alternating, N-acetylglucosamine, N-acetylmuramic acid. Then the next, N-acetylglucosamine, N-acetylmuramic acid. Now, that is the structure of peptidoglycan. And peptidoglycan is the major component of bacterial cell walls. So if peptidoglycan is the major component of bacterial cell walls, and lysozyme acts on the glycosidic bonds between the monomers, breaking down these glycosidic bonds, then lysozyme will have such antimicrobial activity, because once it breaks down the cell walls of bacteria, those bacteria, of course, that is going to result into their death. It's a hydrolytic reaction, which means water, comes in that reaction, aiding in the breakdown of those glycosidic bonds. So we see lysozyme is acting in that process where you have a glycosidic bond between N-acetylmuramic acid and acetylglucosamine, which is broken. And so you now have on that N-acetylmuramic acid, the original hydroxyl group. And then you have also on N-acetylglucosamine, also you have the hydroxyl group there. So water comes in that reaction, lysozyme acts and breaks down that glycosidic bond between these two monomers. If you look at it, zoom into it more, we see, so in the active center of lysozyme, there are two amino acids there. Aspartic acid, 52. Already you know what that number means. Glutamic acid, 35. Now these two will have some hydrogen bonding. So we see hydrogen bond residue, hydrogen bonds to residues of, in the binding site. So in the binding site is the aspartic acid and the glutamic acid that are having some hydrogen bonding on, one of these hydroxyls. That's going to be on carbon number 3, and with the N-acetyl group, which is on that N-acetylglucosamine residue. And you see the cleavage sites, that is that glycosidic bond, which is to be broken between the N-acetylmuramic acid and N-acetylglucosamine. You see it's, zoom in there so that you see lysozyme. So water molecule coming in that reaction, which breaks the glycosidic bond between N-acetyluramic acid and acetylglucosamine to have the separate monomers. So we see that occurring with lysozyme breaking down that glycosidic bond in peptidoglycan, which will result into bacterial death. So that's the action of lysozyme. You find lysozyme is used, as an ingredient in some antibiotics. Yes, it's even used in the food industry for food preservation. So it is from the knowledge that lysozyme can break down the cell walls of bacteria, and so it's used in such applications. All right, so this brings us to the end of what we had to do today. I know I've given you enough headache with some of these, the very last reactions. So now the floor is open. If you have anything to say, you can raise your hand. I will unmute you. I will enable your mic, and then you can unmute yourself and you speak. Alternatively, you can write in the chat, and then I can respond to it. The floor is open.